Hi guys, it's Mr. Y. Today we're going to be talking about the different types of RNA. So our learning objectives for today are learning about the types of RNA and how these types of RNA do different jobs even though they all interact with each other for the same overall purpose. So let's begin. So there's variations on RNA, and there's actually more variations that I'm going to be talking about today, but these are the important variations that you're going to need to know for genetics. These varieties all include the same basic nucleotides of A, U, C, and G. And you recall from our base pairing practice that A will be always base pairing with U, and C will always be base pairing with G as long as you're dealing with pairing between two RNA strands. Remember, it changes slightly if you pair it to an RNA from an RNA strand to a DNA strand. So the first variety we're going to talk about is called mRNA, and you'll notice that the M here is lowercase. That's on purpose, whereas the RNA is capitalized. The M stands for messenger RNA. This is a short chain of nucleotides, RNA nucleotides, that's made when transcribing DNA, and we'll talk about what transcribing DNA means later on in a different lecture. The RNA of this variety you are actually already familiar with. It's just a single strand, not bundled up in any case, of RNA, and each 3 base pairs is called an RNA codon. So here you have A, U, C is one codon, then G, U would be another codon, and then A, C, U would be a third codon, and then you would continue down the line. They're usually much longer than this. I'm just showing you this amount to save some space. So this is mRNA, messenger RNA. The second type of RNA is called tRNA, and again, the T up here is lowercase on purpose. The T stands for transfer RNA, and this is a folded chain of RNA, and it has 2 unique features. It has one free, what we call anticodon, and one attached amino acid, and sometimes they'll call this the anticodon site as well. Looks kind of like this. Now, don't let the picture scare you. If you look closely, though, you'll see that there's C's and G's base paired together, then A's base paired with U's. And occasionally you do see weird little things happening, like a U base pairing with a U, but most of the time it's still A's, U's, C's to G's. And you can see there's base pairing happening here, 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 and then also where the circle already is, here. And the 2 interesting features we talked about, the free anticodon site is way down here towards the bottom. It's just these 3 base pairs, in this case G, C, C. And at the opposite end, at the very, very top, is an attached amino acid group. And here's another picture. Same thing, the anticodon site down here. In this case, the anticodon is AAG, AAG, and then the amino acid would be up here in this section. They're not showing it in this picture. And again, the base pairing sequences should be CG, AU. I'm not sure why that one on the left shows UU. It shouldn't show that in many cases. So these 2 pictures are probably a bit simplified, but they show the same basic idea. And then if you're going to copy any pictures of tRNA down in your notebook, I'd get the one here on the right. This one. Again, it shows you the anticodon here and then the amino acid, which it has a label right here for this guy. So there's the amino acid, there's the anticodon site. And then the rest of it is just regular RNA. In some cases it's bonded to itself, in some cases it's not, where it's too far apart. That's what the dotted lines represent. So tRNA allows for the combining of one specific amino acid, again the amino acid at the top, for one specific type of anticodon. So every anticodon, CGG or CGC or whatever, gives a specific sequence that can bind to a specific, meaning the amino acid sequence, the amino acid on top will be specific as well. So these 2 are related to each other. If the sequence changes, you can get a different amino acid. So they are very specific to each other. Specific. So in comparing these 2 types of RNA, Again, the mRNA is long, stretched out. It doesn't bind to itself, typically speaking. It can bind, mRNA can bind to DNA, whereas the tRNA mostly binds to itself in some cases where it's close together, like here or up here. And it has the two unique features of the anticodon site and the amino acid at the other end. Kind of looks like a T too, if you want to imagine it like a weird kind of off-center T. So that's one way of remembering tRNAs. So mRNA is used in this process called transcription, and again, every 3 base pairs is called an mRNA codon. tRNA is used in a different process that has a very similar sounding name. It's used in a process called translation. Again, that's not the same thing as transcription, so don't get the two confused. We'll talk more about those in a different lectures. And again, it has the amino acid and it has the anticodon site that's specific to that amino acid. Now, this last picture, I would like you to make sure you get down in your notes. Now, you should have this portion in the square up here down someplace already. So if you don't want to get that, if you already have that down, that's fine. But this big picture here, take a few minutes, copy it down. This will be the last slide for this lecture. This is going to be a short lecture. But what it does show is how you get tRNA to interact with mRNA. So the mRNA labeled here is this long black strand here. And then you can see every 3 nucleotides is a codon. And you can see the tRNAs, there's 3 of them in this case. You have one up here, 1, 2, 3, will sometimes attach to the mRNA. And they do this with the help of this shaded thing here, and in the back area here, this thing is a ribosome. You remember ribosomes from the organelle unit? These are the little factories of the cell. These things make proteins, and this shows how they do that. They take the mRNA and they kind of feed it through themselves like a reading tape. And as they feed through, they are reading it, and they are forcing the mRNA to attach to the specific tRNA. And you can see the binding process happening here. And these pairings are, once again, based upon the nucleotides. So A's and U's, C's and G's, because this is RNA to RNA. And as I told you, the sequence here is going to determine which amino acid you get. So you get a very specific set of amino acids, building up, because this chain is going to hop along to the next amino acid, and as the next one comes in, it's going to hop along here, and the chain is going to continue to grow outwards. As the mRNA gets pulled to the left here, there's going to be more tRNAs building, and this chain is going to grow longer and longer. Now, this chain of amino acids, they call it a growing polypeptide, composed of peptide bonds and amino acids. That's eventually what's going to make up a protein when it folds in onto itself. So this is how cells make proteins, by using a combination of mRNA and tRNA and ribosomes in this whole process. So again, if you need to go back to the beginning without all my lovely handwriting on the picture and make a copy of this picture, please do that, because this is a process you will have to understand for genetics. And the title of the process right here is called translation.